Well, hello. Welcome to another lesson of A&P with Dr. H. Today we are going to be talking about the intrinsic and extrinsic controls of autorhythmic cells. So in essence, you got to have a bit of a background before we can start to run through this particular lecture. So let's try to just do a quick review of what we already know about the heart and about cardiophysiology. I will do this review almost every single time that I teach about cardiophysiology because it's a critical background of how the heart works. Write these sorts of things down every time you go to study this. It's going to make it a lot easier, I promise. So what do we know? All right. Well, we know we're studying the heart. And so I recommend that you start there. Just write down heart and say, what's the function of the heart? What do we know the function of the heart is? Well, we know the function of the heart is to pump blood, and more importantly, it's to pump blood over time, right? We want that heart pumping blood through our entire life. And so we can actually define this as something called cardiac output, CO. So the function of the heart is to create cardiac output. And that's what we're after. Well, what about the anatomy of the heart allows for the function of the heart? Well, if you'll recall, the heart is made up of two major cardiomyocytes. One, we have the autorhythmic cells. And two, we have the myocytes themselves, the myocardial cells. And those myocardial cells are going to contract and relax, whereas the autorhythmic cells, they're going to create their own rhythm. They're going to create their own action potentials. So we can define the function of the autorhythmic cells as these are the cells that allow the heart to have an action potential. They have electrical conduction through the heart over time. In other words, these are the cells that create the heart rate. Versus the myocardial cells, which are the ones that are going to contract and relax. And every single time they contract, they create pressure inside of the chamber of those hearts. And that's going to allow blood to be ejected out of the heart in a single heartbeat. And we define this as stroke volume, or SV. And when we take and look at all of this together, well, these two anatomical features of the heart are going to determine the function of the heart. So in other words, cardiac output is equal to heart rate times stroke volume. I cannot emphasize the importance of knowing this equation. I cannot emphasize knowing the importance. Cannot emphasize enough knowing the importance of this equation. Let me say it again. I cannot emphasize enough the importance of knowing this equation. If you're learning cardiophysiology, you need to learn this equation. Because how is the body going to go about regulating cardiac output? Cardiac output is based on the body's demand. So what happens if oxygen demand increases out in a tissue somewhere? What do we want to do to cardiac output? We want to provide more blood out to that tissue, right? So what would we want to do? We'd want to increase cardiac output. Well, what two variables control cardiac output? Heart rate and stroke volume. And what two cells control heart rate and stroke volume? Autorhythmic cells and myocardial cells. Hopefully that helps tie all that together for you. So now if you want a scenario where we want to increase cardiac output, what can the body do? The body can either increase its heart rate, or the body can increase its stroke volume, or it could do both, right? And either one of those is going to increase that cardiac output. So hopefully you guys were able to follow that. That's from previous lectures that I've done, but that's just a real quick review. So what we're going to go in today is how the body goes through and controls the heart rate, because we're going to focus primarily on the autorhythmic cells here, and in the next lecture we'll focus on what the stroke volume and what the myocardial cells are doing and how they affect cardiac output and how the body can regulate them. Right? So if we're going to go about changing heart rate, well, there's a term for that. Of course there is, right? In medicine, there's a term for pretty much everything. So what we want to ask ourselves first and foremost is how, right? We always want to try to ask ourselves how when we're doing physiology. How does something happen? Well, if we're going to change the heart rate, The term for this is a chronotrope. Okay, chronotrope or a chronotropic agent, where chrono means time. Trope is telling you it's a chemical agent. All right, this could be a pharmaceutical drug, recreational drug, or something inside your body like a hormone that is going to trigger an increase in the heart rate based on long-term effects. So chronotropic agents can either be positive chronotropes or negative chronotropes. Okay, we could either have positive chronotropes or we can have negative chronotropes. Again, I'm just introducing some ideas to y'all, then we're going to go through and talk about the actual how. How does this all work? Okay, some great examples of positive chronotropes are ones that you guys probably can already envision, right? What happens when you're running away from Mr. Axeman? When you're in the middle of a fight or flight response, what's your body going to do? It's going to release norepinephrine and epinephrine. And what's that going to do to the heart rate? Positive chronotropes are going to increase that heart rate. All right, so let's list off some of those chronotropic agents, those positive chronotropes. Again, norepinephrine, epinephrine. Those are great ones for you guys to know about. But what else is out there that can increase the heart rate? Well, inside the body, your body actually secretes a really important group of hormones called T3, T4, right? These are thyroid hormone. And what do we know the function of thyroid hormone is? It's to increase the number of beta-1 adrenergic receptors on the cell membrane. In other words, it increases or upregulates beta-1 adrenergic receptors throughout the entire body, including the ones found on the heart. The more beta-1 adrenergic receptors, the greater the degree of sympathetic activation. Nice little correlation there. What else do we know can act as a positive chronotrope? There's quite a few other things. So let's list off, let's go with nicotine next. Nicotine. And nicotine acts on all nicotinic receptors. And if you recall, nicotinic receptors are found on the postganglionic neuron for the entire autonomic nervous system. So this is going to activate both the parasympathetic nervous system and the sympathetic nervous system. But which one do you guys think wins? The sympathetic nervous system always wins because survival right now is more important than digesting your food or resting. So nicotine will be a positive chronotrope as well. One of my favorite chronotropes, positive chronotropes, is caffeine. Caffeine acts by increasing a secondary messenger inside the cell called cyclic AMP. And we're going to be talking about cyclic AMP as we go through these extrinsic controls of autorhythmic cells in just a minute. There are a number of other ones like cocaine. Cocaine decreases norepinephrine and epinephrine reuptake into that terminal bouton. So it's going to allow norepinephrine and epinephrine to bind longer to the beta-1 adrenergic receptors, so forth, so on. So there's quite a few different positive chronotropes out there. Negative chronotropes, what are they going to do to the heart rate? They're going to decrease the heart rate, right? They're going to do the opposite. And hopefully you guys can already start to think of the most common negative chronotrope in the human body, and that's going to be related to the parasympathetic nervous system, right? And that's going to be acetylcholine. So acetylcholine is going to be the major neurotransmitter released by the parasympathetic nervous system for rest and digest. There are other ones as well, especially pharmaceutical drugs that we use very often, like beta blockers. Beta blockers, as the name implies, they block beta adrenergic receptors. There are specific beta blockers, specific for beta 1, beta 2, beta 3, and then there are these, blanket inhibitors, these blanket beta blockers that can block all the beta adrenergic receptors in the body. You'll see how this will impact heart rate once we start going through everything else. But what you'll notice is that the primary chronotropic agents, the primary controls of heart rate, have been ones that you guys have learned about before. Be it the sympathetic nervous system or the parasympathetic nervous system? So we're going to tie that back into what we wrote up here because we want to try to tie all this together. So if we're thinking about autorhythmic cells, we can see that autorhythmic cells can either have an increase in their heart rate or a decrease in their heart rate. Pretty easy connection. But how, right? Always ask yourself how. And if you'll do that, you'll say, if a chemical is going to have an impact on a cell, that means that cell is a target cell. And what do we know about target cells? All target cells in the human body must have receptors for that chemical in order for that chemical to have an impact. In other words, in order for us to have a positive chronotrope, norepinephrine and epinephrine, act on heart rate, well, what sort of receptors are we going to have on autorhythmic cells? We've already discussed this, but now we're writing it down. We know that norepinephrine and epinephrine are going to bind to and activate, that's A with an arrow up, activation, beta-1 adrenergic. Right? That's what we know. So in order for a positive chronotrope to have an effect on the heart, it must have a receptor. What about the negative chronotrope like acetylcholine? Well, acetylcholine now is going to be released from the parasympathetic nervous system. And what receptor is that going to act on? That's going to act on muscarinic receptors. To be specific, it's actually going to activate a mu2, mu muscarinic. But we're going to go ahead and just write out muscarinic here. And if your professor is asking you guys to know mu2, well, now you know how that's tied into this. Okay, beta-1 adrenergic and muscarinic receptors. Excellent, awesome. How do these tie in? Again, this is going to be the sympathetic nervous system. This is going to be the parasympathetic nervous system. Those are going to be the primary controls that the body has in order to regulate your heart rate right now. If you get out of your chair, you need to increase cardiac output to feed more blood to your brain. What's going to do that? Sympathetic nervous system. What's your body going to do? It's going to release norepinephrine onto beta-1 adrenergic receptors, and that's going to increase your heart rate. Boom, right now. How does it do this? Well, in order to understand how the body goes through these extrinsic controls of autorhythmic cells, we just need a brief review on how an autorhythmic action potential is created in the first place. Okay, so when you're looking at an autorhythmic action potential, we've already learned this. Resting membrane potential is negative 60 millivolts. The threshold potential is negative 40 millivolts. The first portion of the curve here is due to the leaking of sodium into this autorhythmic cell. Okay, so we're going to have a whole bunch of sodium. Sodium is always higher outside of the cell relative to the inside of the cell. And this channel right here was called the funny channel. And as long as the funny channel is open, sodium gets to leak into the cell. And if sodium is leaking into the cell, what charge is coming with it? Positive charges. And that's going to start the depolarization of the membrane that you can see down here. And at about negative 50 to negative 55 millivolts, that funny channel kind of loses effect. It's just the very beginning of the leakiness of this membrane. What's going to come into effect next is a transient calcium channel. So outside of the cell, we have a lot of calcium, right? Calcium is higher in the ECF than it is in the ICF. Lots and lots and lots of calcium out here. This channel, this portion of the curve, is controlled by a leaky calcium channel called a T-type or a transient type calcium channel. And as long as it's open, calcium gets to leak in. And once again, what do we know about that? If calcium leaks into the cell, we're going to have some positive charges that come with it. And that is the second portion of this autorhythmic action potential here, this pacemaker potential right here, that is going to allow that cell to continue to depolarize until it hits threshold. But what's going to happen at threshold right here? Well, threshold, remember, this is the energy required to open up a voltage-gated channel. Well, what voltage-gated channel do we find on the membrane of an autorhythmic cell? And the answer is, let me make sure I put an arrow so you guys can distinguish these. The answer is an L-type, or long-lasting type, an L-type calcium channel, one of the most important channels actually in a lot of systems, including the digestive system. Calcium channel here, but very, very important in cardiac function. This is a voltage-gated channel, and there are a lot more of these voltage-gated channels than there are T-type. T-type is just a slow leak, whereas when the L-type calcium channels open, it's going to allow for calcium to flood into the cell. There's going to be a lot of calcium now. And when a lot of calcium comes in, we get a lot more positive charges that are entering the inside of that cell. And that's when you see this spike in that depolarization, this rapid depolarization phase. But at the peak of the curve, depending on what textbook you look at, there's different peaks, +5 to +30 millivolts. I'm not going to write anything over here. It's up to what your textbook and what your professor is teaching you. This L-type calcium channel is going to close, at which time we need to repolarize. And what's the typical way that we repolarize a cell membrane? Potassium efflux. Okay, potassium efflux. Potassium efflux is the most common way for us to depolarize this cell, because what's going to end up happening as we move potassium through a voltage-gated potassium channel, well, potassium is a positive charge. So what's happening to the inside of the cell is positive charges leave. The inside of the cell is becoming more negative. And that is going to allow all these positive charges to rush out and to repolarize that cell. Awesome. Hopefully that was a nice little review for you all. This is a really amazing little, the way that the body is able to auto-regulate itself is just mind-blowing. I was lucky enough to learn about this when I was in 8th grade, and it's stuck with me since then. Lucky enough, quote unquote, because I have a heart condition, and my heart condition has to do with these leaky channels. So let's now go back and say we have normal. But what if we want to increase the heart rate, and what if we want to decrease the heart rate? Well, now we need to change normal somehow. Hopefully that makes sense to everybody, right? And we started to introduce this over here. We had one piece of the puzzle here, and now we have another piece of the puzzle over here. How do we increase the heart rate? Well, what do we know? We know that norepinephrine and epinephrine can increase the heart rate. So write that down. We know that norepinephrine or epinephrine can increase the heart rate. So let's start there. This chemical, neurotransmitter, neurohormone, whichever one it's acting as at a time, is going to move to the cell membrane. And what's it going to do to the cell membrane? What do we know? A target cell has to have receptors. So what we have here is a 7-membrane spanning protein. This is a GPCR. Hopefully you guys have been taught about GPCRs. And this chemical is going to bind to the GPCR. And on the inside of that GPCR, there is a G protein sitting here. Okay, and that G protein could either be stimulatory or inhibitory. This G protein that is going to get acted on by norepinephrine and epinephrine binding to that GPCR is a stimulatory G protein. Okay, so it's a GS. It's a stimulatory protein. And that stimulatory protein gets activated. In fact, what's going to end up happening is GTP displaces GDP, and that's going to allow for the activation of this G protein. And this G protein is going to travel, let's go ahead and go up here. So this G protein is going to go to the cell membrane, nearby cell membrane, and it's going to act on one of my favorite enzymes found inside of that cell membrane called adenylate cyclase. So it's going to act on this enzyme called adenylate cyclase. Let's go ahead and draw an arrow over here. Adenylate cyclase is abbreviated AC. And I don't really have a lot of room, so I'm going to go ahead and just write AC. You guys can write in adenylate cyclase if you have that room. All right. So again, GTP is going to go and displace GDP, and that's going to send this now activated G protein over to adenylate cyclase, where it activates that adenylate cyclase. So it's going to have a positive effect on that adenylate cyclase. Now, what does adenylate cyclase get to do? So cool what it gets to do. It gets to take ATP, which of course, that's not how you do a T. There we go. It's going to take ATP, which of course is the energy of the body, and it's going to take and hydrolyze that ATP into cyclic AMP. This is a secondary messenger. Hopefully you guys have learned about this secondary messenger at this point. Cyclic AMP, pretty much my favorite secondary messenger, because this is what caffeine acts on, and I am a caffeine addict. Yes, even with a heart condition, I love my caffeine. Caffeine stops the degradation of cyclic AMP. So caffeine causes cyclic AMP levels to increase inside of the cells of your body. And what you're going to see is that cyclic AMP as a secondary messenger gets to go activate downstream proteins like protein kinases. More specifically, protein kinase A, PKA. Protein kinases, whether your professor asks you to know whether it's a kinase A or not, I don't know. But kinases in general have one really important function. Kinases in general do something called phosphorylation. And phosphorylation is to take phosphate groups, available phosphate groups, typically from ATP, and it's going to start to tag other proteins with those phosphate groups. Okay, so PKA is going to go around and it's going to start to add phosphate groups to the channels that are up here on the cell membrane in our drawing. And when it does that, I'm just going to write in phosphorylation. It's going to increase the rate of phosphorylation. And when it does that, it's going to increase the permeability of these ions into the cell. So what's that going to look like on our curve down here? I'm going to start out by drawing it in pencil. But if we increase the permeability of sodium into the cell, doesn't it stand to reason that positive charges enter this cell faster? So what just happened to our rate of depolarization? It just increased. If we increase the permeability of this cell to calcium, now calcium gets to enter faster as well, right? What did that just do? We just reached threshold potential faster. We increased the rate of depolarization. What happens at threshold? The voltage-gated calcium channels. The L-type calcium channels are going to open. Now, an important point, I always try to point this out to my students, is that action potentials are all or none. We don't change our action potentials very easily in the body, so they should all look identical. Okay, so you want this action potential to look identical to this action potential. Don't squeeze them together. Don't try to say that the action potential itself occurs faster. No, 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 no. Only the rate of depolarization occurs faster. Now, since the L-type calcium channel is also phosphorylated, and all of these things are leaking a little bit faster, what ends up happening is that this cell doesn't return to resting membrane potential. Instead, it's going to start to depolarize almost immediately. And that funny channel, much more rapid rate of depolarization, The T-type calcium channel, more rapid rate of depolarization. So what just happened? We just hit threshold again already. And when threshold happens, an action potential happens. And then what happens? Boom, the same thing over and over and over. So what just happened to the heart rate? The heart rate just increased by quite a large amount. So instead of 3 beats occurring in this period of time, we now have 4 beats occurring in this period of time. So now let's go ahead and color this. I just find this efficient. Okay. Now we can see it a little bit better. And this is all due to the increase in cyclic AMP inside of that cell. If we can figure out a way to increase cyclic AMP, we can bypass this entire beta-1 adrenergic receptors. And I just realized I never actually labeled this beta-1 adrenergic, did I? So let's go back and do that. We got beta-1 adrenergic. It's GPCR down here. If we can find a way to bypass that, well, we can do that with caffeine. What if we have more nicotine in the system? Well, now that the postganglionic neuron is going to release more norepinephrine and epinephrine. So that is going to activate this GPCR at a greater level. What about T3, T4? Well, now we upregulate the number of beta-1 adrenergic receptors in that cell membrane. Makes it, again, more sensitive to the norepinephrine and epinephrine in the system. Hopefully you guys can see how these are all positive chronotropes now and how the sympathetic nervous system is going to be the key behind our understanding of this. What about cocaine? Cocaine decreases the reuptake of norepinephrine and epinephrine in the terminal bouton, so norepinephrine and epinephrine stay longer at that synapse. So norepinephrine and epinephrine stay bound to beta-1 adrenergic receptors longer, and that's going to cause a lot of positive chronotropic effects. Awesome. What about negative chronotropes? Well, we know acetylcholine is going to be our key to negative chronotropes, right? Parasympathetic nervous system. So let's go ahead and throw some acetylcholine in the system. And what happens with acetylcholine now is acetylcholine binds to another type of GPCR. So it's another 7-membrane spanning protein. On the inside, we're going to have another set of G proteins. This set of G proteins are both going to have an inhibitory and an excitatory effect. Really, the G protein that's on the inside here, they're all inhibitory G proteins. There's an alpha, and then there's a beta and a gamma, and I'm just going to go ahead and put those together. So this is going to be an alpha. It's an alpha inhibitory, so it's actually an alpha I. And then this is going to be a beta and a gamma I. But I'm not going to write that in here. Again, if your professors ask you guys to know that, now hopefully you know a little bit more about muscarinic G proteins. So these are muscarinic receptors here. So when the acetylcholine binds, it's going to yet again activate this set of G proteins. And these G proteins can go and do specific things. So this alpha-1 regulatory subunit, well, that is going to go, let's see, where do we want to do this? Let's go ahead and do that over here. That's going to go talk to a relatively unique set of channels. These are transient potassium leak channels. So these are going to be leak channels that are only open for short periods of time, and we can regulate those using this alpha-1, not alpha-1, sorry, alpha-I subunit. And this alpha-I subunit is going to actually have a positive effect on these potassium leak channels. So in essence, I just end up writing these down as T-type transient type potassium leak channels. They're not voltage gated this time. They're just leak channels. But what do we know about potassium leak channels? Where is potassium higher? It's higher inside the cell, just like we set up here. So if I end up taking and opening up a channel just for a short period of time here that allows potassium to leak out of the cell, well, what charges just left the cell? Positive charges yet again. So what's going to happen to the inside of the cell or along the cell membrane is it's going to become more negative. What do we call that? We call that hyperpolarization. Very important term. Hyperpolarization, because what does this term tell us? It tells us that the resting membrane potential not the resting membrane potential, sorry. It tells us that the membrane potential just got below resting membrane potential. So as potassium leaves, what's going to happen to our action potential down here, right? Well, the action potential is going to be much more difficult to create because as potassium leaks out, we hyperpolarize. We just got further from threshold as we opened up these T-type potassium leak channels. We just got further from threshold. So it's just going to become that much more difficult to create a threshold potential. What else is going to happen here? These subunits, okay, these beta inhibitory and gamma inhibitory subunits, they are actually going to have an effect on, you know what? Rewind, okay? I apologize, my brain, I just got a little brain fart here, but it's not the alpha inhibitory that is going to have an effect on the T-type potassium leak channels. It's actually the beta and gamma inhibitory subunits. I apologize. Never made a mistake before in my life, I swear. Sometimes I just get talking real fast. All right, so we're going to try to cross that out. So it's the beta inhibitory and the gamma inhibitory subunits that are going to have an impact on the T-type potassium leak channels. Again, it is the beta inhibitory and the gamma inhibitory that are going to actually have a stimulatory effect on these potassium leak channels that cause the hyperpolarization. This alpha inhibitory subunit, that's going to go and talk to, and let me use a different color for this so that we can have it just stand out a little bit more. This alpha inhibitory subunit, that's actually going to go back all the way to this pKa. And it's going to start to inhibit the pKa, and as it does that, it's going to take away phosphorylation, right? It's going to take away phosphate groups from all of these channels. And as it takes away those phosphate groups, what's going to end up happening to the speed that sodium can go through the funny channel? What's going to happen to the speed that calcium can go through that T-type calcium channel? They're both going to slow down. So what's going to happen to the rate of depolarization? Well, this slope is going to make it, right? The slope becomes less steep as sodium leaks through slower, as calcium leaks through slower. But now we do hit threshold. And wherever we hit threshold potential, we have an action potential. And that action potential is all or none, so it's going to end up looking identical to the other action potentials. But now everything slows down. We hyperpolarize. We have a slow rate of depolarization. Then we finally hit that threshold potential. And what's going to happen? Those L-type calcium channels open. Then the voltage-gated potassium channels open. And we slow down, we're hyperpolarized. So this here probably is going to be best represented with the color green. Let's make it stand out. We hyperpolarize, and we slow down the rate of phosphorylation on the funny channel, so they depolarize slower. Then we slow down the rate of phosphorylation on those T-type calcium channels, so they depolarize slower. Action potential is all or none, so it looks identical. What did we just do to that heart rate? We slowed it way down. So now, where we once probably had, what, 4, probably even close to 5 beats in this period of time, whatever this interval is, now we only have 2. So 2, what do we know? This is going to be something like the parasympathetic nervous system. What do we know about the yellow? The yellow is going to be the sympathetic nervous system. The multicolored one, that's just normal. That's going to be your normal activation. All right, well, this is both the intrinsic and extrinsic controls of that autorhythmic action potential. The intrinsic controls are the normal activity, right? So now as all these positive charges come in, where do these positive charges go? They go to the next cell. They go through the conduction pathway, right? They're going to go through these gap junctions, this combination of gap junctions and desmosomes. This is where the intercalated disc comes from. But as these positive charges move over to the next cell membrane and depolarizes that next cell membrane, it travels through the conduction pathway of the heart going on to the myocardial cells. So those are the intrinsic controls of autorhythmic cells versus extrinsic. How does the body go about regulating the speed that that heart beats? It's going to end up using the sympathetic nervous system and the parasympathetic nervous system. How do those impact? Let's do this just one more time. Norepinephrine or epinephrine are going to bind to beta-1 adrenergic receptors. Those beta-1 adrenergic receptors are GPCRs with a G stimulatory protein on the inside. A G stimulatory protein, once activated as GTP, replaces GDP. That G stimulatory protein is going to go stimulate adenylate cyclase. Adenylate cyclase is going to increase the hydrolyzation of ATP into cyclic AMP, and cyclic AMP is the secondary messenger that is going to increase the activation of protein kinase A. Kinases phosphorylate things. Going to phosphorylate funny channels, T-type calcium channels, even L-type calcium channels, and it's going to increase the rate that those ions can pass into the cell. Therefore, it's going to end up increasing the rate of depolarization. Versus something like acetylcholine, which now binds to a muscarinic receptor. This muscarinic receptor has a G protein complex on the inside. It's got alpha inhibitory, beta inhibitory, and gamma inhibitory. The beta and gamma inhibitory subunits are going to go, and they are going to basically inhibit an inhibitor, which is going to allow for this leak channel, this potassium leak channel, to open up just for a short period of time at the very beginning of this pacemaker potential. And potassium leaks out, hyperpolarizing that cell membrane. The alpha inhibitory subunit is going to go, and it's going to inhibit pKa. Therefore, less phosphorylation of funny channels, less phosphorylation of T-type calcium channels, and less phosphorylation of L-type calcium channels. So what's going to happen then? Well, now calcium and sodium can enter more slowly, which levels off the slope of that depolarization. In other words, it decreases the rate of depolarization, thus hitting threshold potential at a later time and slowing that heart rate down. All right, well, hopefully this was a good review and a good introduction into the extrinsic controls of autorhythmic cells. In the next lecture, I will be talking about the extrinsic controls and intrinsic controls of stroke volume. Thanks for joining me today. Humankind be both. See you in the next video.